It's very interesting now that in proteomics we can start to attack also the interactive proteomics, the interactions between the proteins. And I think it's very important. It's actually one of the keys of understanding human biology is to understand the interactions between proteins. And obviously here you can attack this with very different methods, which is very good. Antibodies, you know where are proteins, and you can actually use them to capture out complexes. Mass spectrometry also allows you to capture out and then analyze what are the interacting proteins. So overall, I think we now have the tools to really do interactive proteomics and try to understand what are the complexes, what are the stable interactions, and what are the transient interactions that are part of signal pathways. So in the Human Protein Atlas project, which is part of the big Human Proteome project, we have then characterized the proteins mainly with antibodies. And in the tissue atlas that we published 2 years ago in Science, we showed that a lot of proteins are present in all cells. Then the cell atlas that was published in 2017 in Science, we showed that a lot of proteins are distributed to several locations in the cell. And this is of course very surprising for us, and we now want to try to understand what this means for human biology but also for human diseases. So I think it's very exciting for all of us that are working with proteins right now. We had the genome project that was finished in 2001, and of course the next phase to understand human biology and diseases is to understand in a more holistic way all the proteins in the human body. So what I would advise young people today which are interested in life science, and that is to move into this new arena of protein research where you can use all these new tools to actually look in a technology and data-driven way on the proteins and then characterize them, and actually in the end that will benefit mankind.